Hello friends, welcome back to my channel. Hope you all are well and doing good. Today video is in continuity with my previous video of gel electrophoresis. ज़्यादातर स्टूडेंट्स में ये कन्फ्यूजन या डाउट रहता है कि क्या जेल इलेक्ट्रोफोरेसिस टेक्निक प्रोटीन्स और आरएनए के लिए भी यूज़ हो सकती है. तो येस, जेल इलेक्ट्रोफोरेसिस कैन बी यूज्ड नॉट ओनली फॉर DNA but also for proteins and nucleic acid. But your sporting medium is different in cases of proteins and nucleic acid. In proteins we use polyacrylamide gel, whereas in case of nucleic acid we use agarose gel. So what I am going to cover today is what are agarose gel, what are polyacrylamide gels, and SDS paste technology that is used for the separation of proteins. Agarose is extracted from a seaweed. It is a linear polysaccharide which consists of repeating units of agrobiose, which comprises alternating unit of galactose and 3,6-anhydrogalactose. In this figure, you can see the structure of agrobiose units, which comprises alternating units of galactose and 3,6-anhydrogalactose. Agarose gels have a very unique property that in aqueous solution below 35 degrees Celsius, these polymer strands are held together in a porous gel-like structure through non-covalent interactions like hydrogen bonding, and this property is termed as gelation. Let's have a look on the properties of agarose gel. In aqueous solution below 35 degrees Celsius, polymer strands are held together in a porous gel structure by non-covalent interactions like hydrogen bonding. Agarose gel is formed by gelation through hydrogen bonding, not through polymerization. Next is your laboratory preparation of agarose gel. Laboratories के अंदर agarose gel prepare की जाती है ड्राई पाउडर्ड agarose को बफर सॉल्यूशन में डालकर. फिर इस मिक्सचर को तब तक बॉईल किया जाता है जब तक आपको एक क्लियर सॉल्यूशन ना मिले, और फिर इस क्लियर सॉल्यूशन को ठंडा होने देते हैं रूम टेम्परेचर पर ताकि जेल फॉर्म हो सके. सो लेट्स हैव अ लुक ऑन द लैबोरेटरी प्रिपरेशन ऑफ agarose gel. In laboratory, agarose gels are formed by suspending a dry agarose in aqueous buffer, then boiling the mixture until the clear solution formed. This is then allowed to cool at room temperature to form gel. Now the next topic is our polyacrylamide gels, which are used for the separation of proteins in PAGE, that is polyacrylamide gel electrophoresis. Polyacrylamide gels are formed by the polymerization of acrylamide monomer, and it is an example of free radical catalyst. Polymerization is the process through which a large number of monomers join together to form a polymer. So let's try to understand the process of polymerization of acrylamide gel. Polyacrylamide gels are formed by the polymerization of acrylamide monomer, and it is an example of free radical catalyst. The process of polymerization is catalyzed by the addition of ammonium persulfate and the base timid. The role of ammonium persulfate and the base timid in the polymerization of acrylamide gels are remarkable and important. Ammonium persulfate, which is an oxidizing agent, provide free radical for the polymerization of acrylamide gel, and timid, which stands for tetramethylene diamine, rate the formation of this free radical from the persulfate. Now the question arises, what are oxidizing agents? Oxidizing agents are those substances that have ability to oxidize other substances by donating its electron and thus reducing itself. ये जो electrons का transfer होता है for oxidation as well as for reduction, it is only for for gaining the stability of the molecule. Now let us try to understand the process of polymerization of acrylamide gels through these reactions. In this reaction, R is considered as free radical species and M is considered as acrylamide monomer. As we all knew that free radical species are highly reactive because of the presence of unpaired electron that need to be stabilized by transferring its electron or by pairing with the electron that is present in the outermost shell of the monomer molecule, thus producing a new free radical species Rm, which is highly reactive, and this process goes on forming a long chain of acrylamide. Now let's start the topic SDS page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is the most widely used technique for analyzing the protein qualitatively, and it is based on the separation of proteins on the basis of their size. जिस मिक्सचर को हमें या प्रोटीन के जिस सैंपल को हमें रन करना है पेज पर, उसको हम सबसे पहले बॉईल करेंगे 5 मिनट्स तक एक बफर सॉल्यूशन के अंदर, जिसमें आपका SDS और बीटा मरकैप्था इथेनॉल होता है. Now the question arises, why we use beta mercaptoethanol and SDS along with the buffer solution. Beta mercaptoethanol reduces the disulfide linkage that holds the protein in its tertiary structure, and SDS, which is an anionic detergent, denatures the protein. Now let us try to understand this process with the help of this figure. In this figure, you can see the protein in its tertiary structure. Acids represent the disulfide linkage that holds the protein in its tertiary structure. Beta mercaptoethanol reduces this disulfide linkage, unfolding the protein in its linear structure. SDS, which provides negative charge, will bind with this protein and thus denaturing it. Buffer solution along with the SDS or beta mercaptoethanol also contain bromophenol blue, which is a tracking dye, and allow electrophoretic run to be monitored, and sucrose or glycerol, which provide density to the sample so that it can easily settle through the electrophoresis buffer to the bottom when injected into the loading well. Now the protein sample, which has to be run, never loaded directly onto the main separating gel. When the main separating gel is poured between the two glass plates and allowed to settle down, a stacking gel is poured over the this main separating gel, and it is that gel which has well and where the sample is loaded. Now let us try to understand the stacking and separating gel with the help of this diagram. In this diagram, you can see the upper portion represent the stacking gel with a pH of 6.8 and large pore size, whereas the lower portion will represent the separating gel with a pH of 8.8 and small pore size. The main purpose of stacking gel is to concentrate the protein into bands, so before it enters the main separating gel. Now the question arises, why stacking gel has a pH of 6.8 whereas separating gel has a pH of 8.8, although the gel is same? The answer to this question is that both stacking and separating gels are prepared in a tris buffer with different pH. For stacking gel, the pH of tris buffer is 6.8, whereas for separating gel, the pH of tris buffer is 8.8. As we all knew that the purpose of stacking gel is to concentrate the protein sample so that it can form a strong band before it enters the separating gel. Here in this figure, you can see the formation of band, but this band formation is not due to the presence of only SDS protein complexes, but also due to the presence of glycinate and chloride ions that comes from the buffer solution. Glycinate ions, which are negatively charged, has a lower electrophoretic mobility rate as compared to the SDS protein complexes, which has lower electrophoretic mobility rate as compared to that of chloride ion. Now, glycinate ions has a very unique property that they can move at the same speed as chloride ion if they are under the region of high field strength. Once the glycinate ions enters the separating gel, they are fully ionized because of the higher pH environment, and it will increase the mobility of the glycinate ions. This interface between the glycinate and chloride ions leaves behind the SDS protein complexes, which are left for electrophoresis on their own rate. The next step in page is a visualization of protein band on main separating gel. As we all knew that in page, proteins are separated on the basis of their molecular size. So smaller protein can pass more easily as compared to the larger protein, as they are retarded by the frictional resistance due to the sieving effect of the gel. Here in this diagram you can see a small protein band near the positive end of the electrode. Being small molecule, bromophenol blue is completely unretarded and thus indicates an electrophoretic front. The last step in the visualization of protein band on main separating gel involves the removal of gel from between the plates, and then this gel is washed in a Comessi brilliant blue solution, followed by washing in a destaining solution overnight. The destaining will result in the removal of unbound dye, thus leaving a clear band of protein on a clear background. I'm ending this video now. Thank you for your all love and support to my channel. Please subscribe to my channel. In my upcoming videos, I'm going to cover cloning vectors, and I'm also going to make some videos on classification covering the basic concepts of classification of chapter 11. Stay with me till then. Good day. Bye bye.